Hi everyone, welcome back. So previously we talked a lot about temperature mainly as a sterilizing agent. We explored dry versus moist heat and tools that we use like autoclaves and pasteurization in different settings to do sterilization, or in pasteurization's case, disinfection. We also talked about cold temperatures as an inhibition factor, but not an effective sterilizing method. So now we're going to explore some other methods that we use to achieve asepsis. One way that we can do that is through filtration. We can, of course, use heat, like hot air, to do sterilization, but sometimes this is less effective. Instead, filtration is a more effective strategy where we are physically removing and separating microbes from the medium that they inhabit, either air or water. So for the air, when we're filtering air, we are often using HEPA filters. These are high efficiency particulate air filters. This is really like a sieve that air is going to flow through, and things that are a certain size are going to become trapped. As they pass through the filter, they get stuck and they're not able to move effectively through it. And so bacteria are typically, between 1 to 10 microns in size. So these cells are large by comparison to viruses. Viruses are very small. We're looking at them being less than 1 micron. And so a single virus is probably not going to be stopped by an effective HEPA filter. But HEPA filters can potentially stop aerosolized liquids from passing through that environment and carrying those viruses. What do I mean by aerosolized liquids? I mean like tiny, tiny droplets of liquid. Like when you cough, when you sneeze, a lot of that matter, it is aerosolized. It is turned into an incredibly fine droplet that becomes airborne, and it can stay airborne for a long time. We'll talk about this more in future chapters, but for now it's important to note that these aerosolized liquids can be effective carriers for viruses and other kinds of pathogens that are like respiratory pathogens especially. And so we can actually stop, in some cases, the viruses from being spread by stopping the aerosols from being spread. Similarly, when we look at liquids, we can also often apply filtration as an effective method of sterilization, where we can literally remove microorganisms of a certain size and larger. So we can remove particulate that is larger than zero point 0.2 microns in size, which is smaller than we can do with air filtration. And this method can be useful when used in tandem with other sterilization practices like boiling water. So we can actually filter water and then boil that water to ensure that anything that might have passed through that filter is now dead. Radiation can actually be a pretty effective sterilizer as well. We do actually utilize radiation as a sterilizing tool. It's predominantly effective at destroying and damaging an organism's DNA. And of course, if the organism's DNA is damaged, that really impacts its ability to reproduce. So this is a really good option for things that have DNA, like bacteria and fungi and viruses. Obviously not prions, because prions don't have any DNA to be destroyed. So there's two main kinds that we can use. We have ionizing radiation. This is radiation that comes in the form of X-rays and gamma rays. And so this is mainly what we're using when we do DNA destruction. We are actually breaking apart DNA molecules. We actually will blast these molecules with this radiation, and it'll make them lose electrons. So it essentially changes the behavior of these atoms at a molecular or an atomic level. And so this has major impacts, and it can lead to the destruction of DNA. We actually use this pretty regularly in the US on sterilizing practices for spices, meats, veggies, and fruits. We can also use this method to sterilize different kinds of medical supplies, a lot of plastic materials and surgical gloves, things that we can't really put in an autoclave or an oven, and it will, be damaged by the heat. We can actually use a different method, this type of sterilization, which doesn't use heat at all. Then we also have non-ionizing radiation. So this is comparatively less powerful, less energetic radiation. It's mainly in the form of ultraviolet light. This will also damage DNA, but it won't destroy it. It will severely alter and damage that DNA, and it can lead to the death of these organisms as long as you use it for long enough exposure. Now, this method of radiation, this non-ionizing radiation, is what we call non-penetrative. It means that it's only effective on the surface of whatever item you are trying to sterilize. So we use this in our lab to sterilize goggles. Because the goggles are a form of PPE that has a distinct surface to them. It's not like a medium that has a lot of like three-dimensionality to that, and microbes are not hiding on the inside of these goggles. If they're there, they are only on the outside. And so this method is a pretty effective one. We also have chemical sterilization. So there's a variety of different kinds of chemicals that exist that can act as sterilizing agents. And the methods that they use to kill or inhibit these different organisms and also their applications vary a lot. So when we talk about these different chemicals, I want you to make sure that you know about the concept of modes of action. A mode of action is just the different method or approach that a chemical has to interfere with a microbe. Some of these chemicals, for example, will alter the permeability of a cell's membrane. It'll make it less able to keep things out, and it'll make the membrane more porous, more leaky, more likely to break apart and lyse. Other modes of action might involve damaging proteins directly. Again, protein function is determined by protein shape, and so we can interfere with that shape to affect their function. We can also use the same approach that we used for ionizing and non-ionizing radiation, where we attack the DNA and the RNA in some way, where we can alter the organism's ability to reproduce or to access their genetic material and make proteins from their DNA. Now, the vulnerability to sterilization is pretty variable across species for these chemical methods. There are some organisms that are much more vulnerable to these types of sterilization methods and some that are much more hardy. So starting at the very bottom here, from least resistant, we have viruses with lipid envelopes. These are pretty vulnerable viruses, all things considered, and they can be very easily killed. Above that, we have gram-positive bacteria. They tend to be much less resistant to different forms of sterilization. Well, above that, we have viruses without envelopes, so we have the naked viruses. Above that, we have fungi and fungal spores. Most fungal spores are really hardy and able to survive a lot. Remember, this is distinct from an endospore. These are not the same thing. Above that, we have most gram-negative bacteria. Gram-negative bacteria, having that extra outer membrane, makes them a bit more resilient against different chemicals they might encounter. Then we have protozoa, so vegetative or just simply active protozoa that are kind of out and about and doing their thing, are generally pretty hardy. Above that, we have cysts of protozoans. So these are protozoans that have, very similar to an endospore forming bacteria, undergone some kind of stress and have triggered their survival instinct where they undergo a cyst formation. Those cysts, just like an endospore, they're really hardy, they're really long-lasting and tough, and they can resist a lot of things that would normally be very effective at destroying a protozoan. Above that, we have a group of bacteria called mycobacteria. Mycobacteria. So this is a major group of bacteria that have a specific kind of chemical in their cell walls called mycolic acid. This is a waxy lipid that has a really resistant quality against a lot of different chemicals. So a notable representative of this group is Mycobacterium tuberculosis, the species that causes tuberculosis. This group is really tough, really hard to destroy, and a lot of our Normal sterilizing or disinfecting agents don't do a very good job at killing it. Above that, we have endospores. Endospores of specific bacteria. Remember that only gram-positives and only certain gram-positives are able to actually form an endospore. And when they actually have made an endospore, they are so resilient that they resist most forms of chemical sterilization. They really need to be killed with heat predominantly. That's the main way that we go about destroying them. And above that, the most resistant things that we know of right now that resist different sterilization methods are prions. We don't really have any good chemical sterilizers for prions. And our methods for prion control are predominantly through heat and other ways that we can deform that protein and make it lose its shape. In the laboratory, we can actually measure the resistance of different bacteria to different kinds of chemical sterilizers. What we do is we grow the bacteria on a plate, and instead of our usual method of streak plating that you've been familiarized with, we want to evenly spread the bacteria out so that they are uniform across that plate's surface. Then we can take these little porous discs and we can soak them in these chemicals of different kinds. And the porous medium of that little disc, these little white discs that we see on the plate here, they can soak up these different kinds of chemicals that we put them with, and we can apply them to the plate. And then we wait and see if the bacteria can survive and if what we call a zone of inhibition forms. A zone of inhibition is just an area around that disc in which the living things that are growing on that plate cannot survive. Either their growth is inhibited, preventing them from growing, or it has outright killed those organisms and left a clean, barren space. Now, how effective that chemical is at killing that bacteria, and conversely, how resistant that bacterium is against that chemical sterilizer, is determined by how large and wide that zone of inhibition is. Now, for disinfection, we also have different kinds of chemicals that can do disinfection. We call them, of course, disinfectants. And just like with chemical sterilizers, different disinfectants can have different modes of action. Some will attack one part of a cell and some will attack another part of a cell. They might attack things in different ways. Remember that these disinfectants, they are not sterilizers. These are not the same thing. A disinfectant is a chemical that is going to remove potential pathogens from a surface. It will not fully eradicate all organisms on a surface. It'll also not treat a disease or deal with a problem that already exists on a person. This is more a preventative tactic to make sure that someone doesn't come in contact with a surface that is contaminated with a pathogen. It is also not something we use to clean a wound. That is only something we use on a non-living surface. The stuff we use to clean a wound is an antiseptic. It's a different type of chemical entirely. So many different kinds of chemicals can be used as disinfectants. However, no single disinfectant is universal. And that, by the way, is a similar phenomenon with chemical sterilizers. There really are no chemical sterilizers that will kill everything. All these chemicals are going to vary a lot in how they attack microbes and how effective they are at killing specific things. And microbes, because they are incredibly different, can vary dramatically in how vulnerable they are to these different chemicals. Then, of course, we have soap. Washing our hands and washing surfaces mechanically. Soap can be a very effective disinfectant, but not necessarily because it's killing bacteria. Soap is an abrasive, and this abrasive and rubbing action that we use when we rub our hands together can help mechanically remove bacteria. Soap also can help disrupt the glycocalyx and the adhesives that bacteria use to stick to a surface like your hands. Soap also qualifies as a type of chemical called a surfactant. So a surfactant is something that will mess with the hydrophilic and hydrophobic qualities of different molecules, particularly lipids. A surfactant will totally mess with and interfere with that quality of the cell membrane, and it can make the cell membrane actually break apart. It can help it actually undergo lysis. So soap on its own is very effective. We can also have different kinds of soap that have antibacterial agents mixed in. So antibacterial soap is just one type of soap, and its effectiveness is actually a bit questionable. Antibacterial soap can actually help cultivate antibiotic resistance in some bacteria, because when we are introducing bacteria to antibiotics, we naturally create a type of selective pressure that organisms that are exposed to antibiotics, the vulnerable ones, are going to die or be otherwise inhibited in some way. And the organisms that happen to have a mutation that lets them resist that antibiotic, they will do fine. Through this exposure, we are filtering these bacterial communities and removing organisms that are more vulnerable to these antibiotics and helping those that are more resistant proliferate. And then bacteria have that frustrating ability to do horizontal gene transfer, where they actively pass genetic information back and forth between each other in different ways. And so these organisms that have evolved antibiotic resistance, it doesn't just stay in the direct lineage of those cells as they reproduce. It is very possible for a cell that has antibiotic resistance to share that antibiotic resistance with a neighboring cell that doesn't have that gene. So today, we really prefer to restrict the use of antibacterial soap to food handlers, newborn caregivers, and immunocompromised patients, because those are the situations where that stuff is really useful, having that extra little edge, however little it may be. But people going about their everyday who are not necessarily in those fields or specific situations probably don't require that kind of killing power. So in healthcare today, aseptic treatment of different kinds of items, it follows something that we call Spalding's classification. This is essentially a breakdown of the priority of asepsis and how we apply different methods of asepsis based on a particular item's use. How are we going to sterilize this tool versus that tool? So critical items are things that require sterilization. These are tools that are going to be used in a medical setting to break the skin, that will break through another membrane, not just the cutaneous membrane, and any items that enter a sterile body cavity. These have to be treated with sterilization tools and techniques. Semi-critical items only require disinfection. So these are tools that are going to be in contact with different membranes, that will not necessarily breach those membranes, but will be in contact with them. And things that are in contact with non-intact skin need to be disinfected. Non-critical items require only minor disinfection or basic cleaning. So this is effectively everything else. Things that are going to be in contact with healthy patients, with healthy skin that is intact and not damaged in some way. This is an important breakdown of priority in terms of what tools receive asepsis in what way and in what priority.